Welcome everyone to the CARPHA COVID-19 Health Rungs webinar on gene sequencing for COVID-19 variants, which you know is a hot topic. Today we're going to be looking at laboratory testing perspectives. And of course, it's for us to understand the evolution and introduction of the COVID-19 variants of concern, to discuss considerations for gene sequencing, even as a surveillance tool here in the Caribbean. And for that, we've invited our experts from the University of the West Indies and from our sister network in Canada, from the Canadian Public Health Laboratory Network. And so we welcome Dr. Christine Carrington and Dr. Gary Van Dommeslaar. This time, I would like to invite Dr. Lisa Indar, Director of Surveillance, Disease Prevention and Control at CARPHA, to bring her welcome remarks. Dr. Indar, thank you, and over to you. We just unmute Dr. Indar. Just let's.
Oh, yeah. I was saying so much nice things before. Let's start again. A very pleasant welcome, everyone. And let me just say welcome to Trinidad and Tobago in virtual. From those, I'm seeing persons from all over the world. So it's really, really nice to have you here. It is my distinct pleasure today to welcome you to this CARPHA COVID-19 Health Rounds that has been organized by the Secretariat of the Caribbean Public Health Laboratory Network, Carrie Flynn, to discuss this very topical, and I want to say crucial issue right now of gene sequencing for the COVID-19 variants. I welcome our colleagues to this in the regional response. You know, the well-renowned Dr. Christine Carrington, colleague and partner and friend as well from the University of the West Indies, and Dr. Gary Van Dommeslaar. Did I say that right?
Yeah, you nailed it.
From our sister network, the Canadian Public Health Laboratory Network. Welcome very much, and thank you for agreeing to participate in this important webinar. From a regional perspective, we know that most of the COVID-19 variants of concern has been identified in the Caribbean, and we have had variants of concern as well as variants of interest, and we are the most travel dependent region, so it's something that is expected. The predominant ones that are circulating though are the alpha, gamma, and more recently, you know, the Delta variant. And this is of grave concern for us because just picture us in the Caribbean, these small communities, small countries, you know, of varying capacity. And, you know, there is that potential that the spread of these variants could have grave impact on not just on the population and illness, but, you know, economic sustainability on the whole. So we do recognize that several of our largest member states are actively involved and engaged in balancing their ongoing laboratory testing response to identify new cases of SARS-CoV-2, while also seeking to introduce gene sequencing as well. Through the CARPHA mechanism, we will continue to work together with our national laboratory members and other key stakeholders to support the development of this capacity within the region to enable the surveillance of the variants of concern with a view to sustainable integration into our existing laboratory surveillance for pathogens of public health importance. We all know the importance of laboratory-based surveillance, and now even more so, gene sequencing is a critical part of laboratory surveillance. So without further ado, I don't want to take up your time. We have important people to speak. I want to wish you all a successful meeting and look forward to the stimulating questions that would be posed to our panelists. Over to you, Sasha.
Thank you very much, Dr. Indar, and thank you for taking the time to spend a few moments with us. We know you have a very busy schedule, so hopefully you stick around to hear a little bit more. So let's go directly to our first speaker. She pulls up her presentation. It's my real privilege to introduce Dr. Christine Carrington, who was one of the more exciting lecturers during my undergraduate degree at UE way back in the early 2000s. And so Dr. Christine Carrington is a professor of molecular genetics and virology in the Department of Preclinical Sciences, the Faculty of Medical Sciences, and University of West Indies St. Augustine campus. She holds a BSc in biotechnology and a PhD in molecular virology from the University of London. Dr. Carrington's research focuses on understanding factors involved in the emergence, spread, and maintenance of emerging viruses. She's currently head of the Department of Preclinical Sciences, member of the University of the West Indies COVID-19 Task Force, and a member of the Technical Advisory Group on Immunization to the Trinidad and Tobago's Ministry of Health. In this regard as well, and as part of the COVID-19 response, Dr. Christine is the principal investigator for the COVID-19 Impact Project, which she'll be talking more about in a few moments, that established local capacity for SARS-CoV-2 genomic surveillance in support of the COVID-19 mitigation and control efforts of local and regional public health bodies. So Dr. Carrington, it's my distinct pleasure to welcome you to this forum, and I hand over to you. Welcome.
Thank you so much. It's a pleasure to be here and to have the opportunity to speak about this project. I'll just share my slides. Are you seeing my slides now? All right,
yes,
great.
Everything is looking great.
All right, so let me just get my little pointer up and I'll be ready to go. So, good morning to everyone, or good evening, or good afternoon, wherever you are. And thanks very much for inviting me to come and speak. As was mentioned, I'm going to talk about the genomic surveillance that we've been doing in the Caribbean as part of a project called the COVID-19 Impact Project, and about SARS-CoV-2 evolution as a prelude to that, So this genomic surveillance, as most of you know, is basically the use of genomic sequencing technology to detect and track the different SARS-CoV-2 lineages that have emerged over the course of the pandemic. So I always like to start with a little orientation. Everybody's familiar with the WHO COVID-19 dashboard. To date, this virus, SARS-CoV-2, is now responsible for over 200 million reported COVID-19 cases and more than 4 million deaths around the world. However, thankfully, we now have several safe and highly effective vaccines. And as of today, more than 4,000 million doses have been administered around the world. Unfortunately, not evenly distributed across the world, but at least we now have vaccines. It's just to get them out to everybody. We know that these vaccines are safe and they're effective, but at the moment, we don't know how long the protection that they offer will last, particularly with the new variants of the virus emerging as the virus evolves over time. So before I go on to talk about the project, I just want to say a few words about virus evolution. Just to orient everybody. So we know that when a virus is transmitted to a susceptible individual, it enters their cells and it replicates to produce millions of new virus particles. And during that process, millions of copies of the virus's genome are made and they're incorporated into new viral particles. And we know that viral genomes can be made of DNA or RNA. And in the case of the coronavirus, these are RNA viruses, and their genome, the genome of the SARS-CoV-2 virus, consists of a single short RNA strand that is 30,000 nucleotides long. And each time a copy is made, you can get errors occurring, and we know that these errors are what we refer to as mutations. Okay, so Viruses in particular, and in particular RNA viruses like SARS-CoV-2 have very high mutation rates, millions of times higher than eukaryotic organisms. And compared to DNA viruses, like this herpes virus shown here, the RNA viruses mutate very rapidly. But compared to other RNA viruses, coronaviruses, which are shown by these stars here, including SARS-CoV-2 in purple, they are at the lower end of this evolutionary rate. So they're fast, they're RNA viruses, so they mutate quickly, but slower than many other RNA viruses. Okay. This figure is just to illustrate, it's a short segment of SARS-CoV-2 isolated from patients around the world, and just to show the different mutations. You know, the vast majority of the genome remains the same, but you do get these mutations occurring. And this is normal and expected for the viruses to evolve and to give rise to multiple lineages, which are basically descendants of the original virus. That have mutations that distinguish them from each other and from the original virus. And on average, SARS-CoV-2 accumulates 2 single nucleotide mutations per month in its genome. So it's you know, you get all these lineages evolving. And each time the virus enters a new person, it goes through rounds of replication in every infected cell. And the longer we know, the longer the pandemic goes on, the more opportunities for replication and the more chance for this evolution to occur. Now, most mutations that occur are actually deleterious to the virus, and the virus particles that contain them do not survive. So for example, here we have 3 mutants in an individual, and mutant 2 is non-viable, so it does not get transmitted to the next person. 2 other mutants, the ancestral one and one that has increased replication, they are transferred. Now, a large proportion of mutations have no effect on the fitness of a virus, and whether the virus containing them survives or not is just a matter of chance. However, occasionally you do get mutations arising that affect the virus's properties. You know, it might affect how easily it spreads, the severity of disease it causes, how well vaccines work against it. We've seen that with the variants. And in some cases, mutations confer a fitness advantage. For example, if we consider this 3rd mutant here, which has acquired mutations that allow it to replicate more quickly, it has a greater chance of being transmitted to another individual. Okay, so it has a fitness advantage over the ancestral virus and is more likely to be transmitted. It's important to note, however, that increased prevalence of a given lineage, a given viral lineage, doesn't necessarily mean that it has a fitness advantage, because there's always an element of chance. Some things can just happen by chance. Right, so now, of course, by comparing the consensus sequences of viral genomes, it is possible to reconstruct the evolutionary history of the virus in a population. And this tree here is constructed from the 3 sequences of the viruses I showed before. And it shows that the 3 of them arose from a common ancestor, which gave rise to this, this virus here, and also then gave rise to a common ancestor for viruses 2 and 3, which are more closely related to each other than to the other ones. So now the reconstruction of a phylogenetic tree is, you know, more complex than just looking at sequences and seeing which ones look most alike, because you have to consider things like whether some changes are more likely than others, some nucleotide changes might happen more often than others, whether some regions of the genome mutate faster than others, and the fact that a series of mutations can result in a change that reverting to the original sequence. So you have to have a model of evolution and a variety of considerations and assumptions when you are reconstructing an evolutionary tree. Now, since the beginning of the SARS-CoV-2 pandemic, scientists have been sequencing viruses in samples from infected individuals and using models of evolution to reconstruct the sort of SARS-CoV-2 family tree shown in this figure here. And each of the colored branches here is a distinct lineage of the virus characterized by certain mutations. And the map on the right here shows the global distribution of those lineages and proportions of each lineage found in different countries. Now, was in January 2021. Now, at the start of the pandemic, all the viruses infecting people were very similar, but over time, they have diverged and branched out to give this family tree that we're seeing here. So you can see increasing divergence and viral diversity across the globe. There are over 1,000 named SARS-CoV-2 lineages at the moment, okay, in circulation. So there's a number of, you can reconstruct evolutionary history of a virus, but that's just the beginning of what can be done with this type of approach. There are a whole range of questions that can be answered through genome sequencing. Some of them are very familiar. So for example, the first gene, you can use the sequence to identify a virus. And at the start of this epidemic, it was genome sequencing that allowed scientists to identify the virus as a coronavirus. Also, due to the rapid mutation rates of the viruses, if sequence data is combined with information on when and where the sample from which it was derived was collected, you can track the spread of the virus. And if virus genome sequencing is undertaken rapidly and on a large scale, as we've been seeing over the course of the epidemic, then it can assist epidemiologists and public health authorities in understanding how the virus is spreading and evaluating how effective their interventions have been. You can look at the rate of epidemic growth of the virus. This can all be inferred from the sequence data. And as we know, it can also help to distinguish whether new variants are associated with particular patterns of symptoms or severity of disease and or rates of spread. And now that we have several vaccines, tracking new variants is going to be extremely important, as we've seen, to ensure that the vaccines remain up to date with the strains of the virus that are currently circulating. Okay. So with all that in mind, very early we proposed that this was in early 2020, we proposed the COVID-19 Impact Project. And the aim of the project was to establish local capacity for rapid generation of SARS-CoV-2 whole genomes. In Trinidad, we had capacity for, I think, Sanger sequencing, which was not appropriate for this type of work that we're doing here, the rate at which it could be done. And we have worked for a long time looking at virus evolution, collaborating with overseas, researchers. And what we used to do was to sort of, do a lot of field work in Trinidad, sample collection, and then for the actual sequencing, we would send those samples overseas to our collaborators. They would send back the sequence data, and then we would do analyses on that sequence data locally. And clearly that was not going to be a reasonable approach to use or feasible approach in the height of the epidemic. So we wanted to establish that capacity. And the aim is to be able to use viral genomics and related molecular epidemiological tools and approaches to incorporate that, to be able to incorporate that into public health efforts aimed at COVID-19 mitigation and control. And this is a UE-led project, UE meaning the University of the West Indies, which is the institution that I am from, which is a regional institution that has, that serves several Caribbean countries. The project is in collaboration with the Trinidad and Tobago Ministry of Health and also CARPHA. And we have collaborators at the University of London and the University of Oxford. CARPHA was instrumental in getting chief medical officers around the region on board with the project, and they were very quick to appreciate the potential and the importance. So we're very grateful for that and for their contributions. Now, the specific objectives of the project were to implement the Oxford nanopore technology. We were very modest. We wanted to use the MinION device, which is this little tiny device here. We didn't have a lot of money to start this, and also to implement the Arctic network pipeline for SARS-CoV-2 sequencing and bioinformatic analysis at our at the university campus in Trinidad and Tobago. We wanted to generate baseline data on SARS-CoV-2 lineages circulating in the Caribbean through the whole genome sequencing. Now, remember, this was proposed back in May, long before the variants of concern emerged. And a key thing is we wanted to be able to respond to public health bodies, requests from public health bodies for answers to questions related to COVID-19 mitigation and control by sequencing samples from the outbreak. And with funding from the Trinidad and Tobago UWI Research Development Impact Fund, we were able to initiate this project in September 2020. The nanopore platform that we chose to use is actually ideal for resource limited settings such as ours. The project, and I'll tell you the project, we started off with $100,000 US, which at the start of the, when we first proposed the project, I had proposed it for, to cover 2 years of work, which that money is the cost of an Illumina MySeq device. And the MinION device, that costs about $3,000, $4,000 US. And although the flow cells are not cheap, the fact that you could pay as you go, use as you need them, that was attractive to us. With this little tiny MinION device, we were able to achieve our aim of implementing the technology, and we generated our first SARS-CoV-2 genomes in December 2020. Now, within 2 weeks of reporting those first sequences, the news of the variants of concern came out, and we very quickly had to pivot from a sort of research mindset to a more service mindset, because the questions that we promised to answer for the public health bodies were we sort of expected to be doing little cluster investigations and things like that. And suddenly we were thrust into this surveillance mode, doing regular reporting, more formal, with lots of, turnaround times being a concern, and, trying to keep up with the demands. Now, for lineage assignment, when we sequence the virus in the samples, we then assign them to SARS-CoV-2 lineages. We use a variety of tools. We rely mainly on the Pangolin software and the Scorpio software, and also the Usher phylogenetic placement software we use quite a bit, among other things. And we also use another range of other online resources to keep up to date with the various lineages, mutations, the variants of concern, and variants of interest. And the sequences we generated, we upload them to the database, which, as many of you know, is a free open access database that encourages open and rapid data sharing, while ensuring that the people contributing the sequences don't forfeit their intellectual property rights to the data. So it facilitates real-time progress in pandemic virus research, and it was first implemented for influenza virus. And it has been an incredible resource throughout the epidemic. For our project, if we detect variants of concern, we only upload them after the countries that have submitted the samples have had a chance to report them to their public. So they have to be public to the local public before we will put them up on GISAID. And we did about 1,000 genomes using a single MinION device, and a lot of very hard work by 2 people, Dr. Nikita Sahadeo and Vernie Ramkasoon in my lab. And then thankfully, recently we got funding from the Trinidad Ministry of Health to buy this GridION device, which does 5 times as much and is also much faster. And we also have 2 additional people in the lab now, courtesy of PAHO, support from PAHO. So things are improved. Now you would be familiar with the variants of concern. So variant of concern is a virus with one or more new mutations that has been shown to spread more quickly or to cause more severe disease, or that affects how well vaccines work. And as we know, there are currently 4 major variants of concern. The WHO labels are alpha, beta, gamma, and delta. And they are each characterized by specific groups or constellations of mutations. In addition to these variants of concerns, there are other lineages of the virus being monitored. Some of them are designated as variants of interest, and others are just listed as alerts for further monitoring. So they're keeping an eye on them. So our project, as I said, we generated our first sequences in December 2020. And as at August 2021, we have received 1,474 samples from 17 countries across the Caribbean. And not all of those samples are suitable for sequencing. Some of them fail the quality control, but we've assigned sequence and done lineage assignment for 1,217 of those. Nowhere near the tens of thousands done in places like the UK, the hundreds of thousands in the UK and all that. But this is, this is what we manage here. The lineages that we have identified in the Caribbean so far belong to we've identified 52 different lineages across the Caribbean so far, including all 4 variants of concern. We have alpha in 4 countries, beta detected in 1, gamma in 7, and now and as of the 1st of August, delta in 5. Trinidad just detected and reported delta virus in quarantined individuals for the first time. So this number is now 6. Right, so you will notice that the majority of sequences come from Trinidad and Tobago. This is because we receive samples not just from CARPHA, but also through the Ministry of Health directly to the project. In terms of turnaround time for this type of work in our setting, if you were to go in front of the machine and sort of put a sample on in the morning, you could get an answer by the end of the day or the next day. So the actual lab work involved with each, physical lab work and is about 1 or 2 days, and 2 days, I would say, working at, not working through the night. And then we have all the analyses to do afterwards and the verification and everything. And so we can potentially turn around a sample in 4 days, but we're not doing 1 sample at a time, and we usually have a lot of them to go through. So we say from the time the samples reach our lab at UE, and remember for the CARPHA member countries, they are going through CARPHA. CARPHA will extract the RNA, do the PCR, and then they send it to us. We try to get them back out within 7 days. And then, of course, it has to go from CARPHA back out. They will verify things, double check, things like that, and then it will go back out to the countries. So it takes some time. We have turned around samples in 1 day if it's very urgent, and 2 days and things when it's very urgent. Right. So this graph shows the number of sequence samples and lineages identified by the week of sample collection. So starting off here in 16th of March of 2020, that's the earliest sample we received. Although the sequencing started in December, we had some retrospective samples. And you'll see that various of the variants of concern have all been detected, and I've shown here the weeks in which each of those samples containing those first variants of concern that we identified were collected. Now, the shape of this curve here does not reflect the epidemic curve across the Caribbean. This is for 17 countries. This is skewed by the fact that most of the samples are from Trinidad and Tobago. And we can see that here. Here we have the epidemic curve that we have. So this is reported cases of COVID-19 in Trinidad and Tobago throughout the over the past this is since July 2020. And for Trinidad and Tobago, we have identified 19 lineages of the virus, including the most prevalent ones we found were B1.111, R1, and then P1, which is the Gamma variant. The B1.111 accounted for about 10% of the sequences we've had to date, and were mainly associated with this time frame here, the early first sort of wave that we had. And then R.1, which is listed as a variant, it's an alert for monitoring, so it's one that's being monitored. R1 accounted for about 24.1% of our sequences, and P1, the gamma, made up about 57.9% of the sequences, and all the other lineages accounted for less than 1%. Now, in terms of community transmission, so the main ones we've seen in the community, remember, many of these samples are captured at in quarantine and at the airport, at the borders. So the main one, this main wave that we've just had, it started off with, if we look here in red here, this lineage here is R1. So during this second, so so R1 was first detected in April 2021. And it was first detected in April 2021, but it was first sampled in February. Here we have P1 that came in after the R1. And as I said, these are the most common. And what we noted is that amongst the samples that were being sequenced, P1 rapidly replaced the R1 as the most prevalent. And today, the vast majority of the sequences that we get are R1 sequences. P1, that's gamma sequences in Trinidad and Tobago. Now, we have done phylogenetic analyses of the Trinidad and Tobago sequences, and it suggests that the R1 and P1 clades associated with community spread in Trinidad and Tobago each arose from a single introduction, with their most recent common ancestors estimated to have existed in the case of R1 around January 2021, and in the case of P1, just at the beginning when we started to see this rise in in March 2021. And we need to do further investigations to confirm whether this replacement that we see among the sequence samples is representative of the general population, and if it's a result of a fitness advantage as suggested by the work done in Brazil on this virus. This is just a slide showing we've done, this is older data when we had fewer sequences. We did analyses on the P1, the subset of P1 samples from Trinidad as the gamma samples, and most of them clustered here. There was this one sample here, which actually is the first one that we detected in Trinidad, but it was from an individual who was on a vessel offshore and who never came onshore. So that was a single one. It never became, was not associated with community transmission. This is the main community transmission here. The P1 sequences in Trinidad are distinguished from other P1 sequences by mutations in open reading frame 1A and open reading frame 3A. So that's just showing what we've looked at there. And of course, we have many more sequences now and want to do this for both P1 and other lineages as well. And just to conclude, the capacity that we have built is applicable to other pathogens, and of course, and it can be expanded to address future emerging diseases, existing diseases of public health importance and agricultural importance. We can apply it to antibiotic resistant bacteria surveillance. So we're very pleased to have this new capacity. Of course, we want to build on it. We still have limited funds. Manpower is an issue. We have people with the with certain areas of expertise, and we can collaborate with, but we need to build our own in local capacity. And of course, one of the issues with the regional surveillance is that issue of transporting samples across the region in under pandemic conditions. And what we would like to see is this sort of capacity being built elsewhere so that we have a more even coverage of the region, the Caribbean region. And just to end, as I've indicated, the study was funded by so far we've received money from the RDI fund and from PAHO. We also have have received in-kind support from the Ministry of Health and from CARPHA. And I just want to thank all the people who have contributed to the project. And this is the team of people in the UE lab who do all of the sequencing. Particularly want to mention Nikita Sahadeo, who's first set up the whole system. And, they've been working very hard to carry out all the analysis, the lab work and analyses. And there's also our collaborators. We have other collaborators at UE, and we have our collaborators at the University of Oxford, University of London, and Imperial College. Nuno Faria in particular, who did all, sort of led the P1 of gamma variant research in Brazil. He is one of our collaborators, and Sarah Hill has been very helpful when we were setting up and troubleshooting. J.T. McCrone has just joined us from University of Edinburgh with some of the analyses. And I'd like to especially thank the chief medical officers from all the CARPHA member countries who have contributed, and we hope that we have been able to contribute to your efforts in your own countries. So thank you very much. I'll end my, right, so thank you very much.
Thank you very much, Dr. Carrington, for an excellent presentation. And I think that those of us who may not know the in-depth parts about gene sequencing and even the timeline that you presented, I think you've really given us an insight as to even introduction of these different variants. Now, we haven't had any, oh, I'm actually now checking the Q&A box. And I think that that maybe you can answer the question by Wesley. First, can you explain the process for selection of samples for further examination?
Okay, so and there's another one, what were the criteria of for choosing the samples? Okay, so as you know, as I mentioned, when we started this project, we had limited capacity. So for the most of the Caribbean islands, we were sort of limited to 10 samples per month, which is not enough to cover, to sample the diversity in those countries, especially if they're having a surge. I think the PAHO recommends that you do random sampling of about 5% of the positive samples. But in Trinidad, where we had a lot more samples coming in, we had a more, the sampling is more controlled. So what we do is 1 in 20, or what we aim to do, I should say, is 1 in 20 of all the reported cases, the reported community cases, random sample. We also receive all of the samples, positive samples in repatriated individuals who are quarantined. So our borders in Trinidad were closed up until the 17th of July. So everybody who came in had to quarantine and undergo state quarantine. So all of those people would have been quarantined for however long, and we would get samples if anybody turned positive. We have also more recently said that we would expect to receive samples from all from reinfections, suspected reinfections, any infections in vaccinated individuals, and any other samples that sort of targeted sampling of things like deaths, people, and things like that. One of the difficulties we've had, and it's because of the way the project started off as a sort of small little thing and then morphed into this massive surveillance, is the marrying the metadata with the sequence samples, the sample sequencing. In order to do, to really know how good our sampling is, we really need to know whether a sample is local, whether the person, whether it's a traveler, where that traveler came from, were they quarantined. We need to know, the date of collection. We usually get that date of onset of illness. There's a lot that we need to get together. And some of that for Trinidad and Tobago is available, but we're still working hard to try and marry it with the sequences, because the whole process was not, it wasn't a seamless transition from a small project to this surveillance, this capacity. So, right.
Thanks very much, Christine, for that. Just want to also note that the criteria for submission of samples from countries through CARPHA, that was shared through the official mechanisms to the ministries of health. So our main labs that we deal with are the National Public Health Laboratories. So they're responsible for getting those samples to us. So they would have that information. The CMOs, the national epidemiologists would have the criteria to help in the selection of samples. And I know that Dr. Carrington and the team, also with our team here at CARPHA, are looking at the current criteria at this time. So for those persons working with us closely in this, do stay tuned. Very quickly, yes, we will be posting the video recording of this webinar so that you can replay it. I know Dr. Christine has said many important things, and that some of us may want to go back and look at and listen again to what she was describing. So that would be made available. Dr. Christine, the question from Neil Phillip, with respect to I don't know if you're seeing it in the
I'm
early
seeing it,
stage.
but I'm not seeing Neil Phillip there. Oh, this is oh, sorry. You know what? I'm looking at the panelists. Okay.
Right. So the Q&A box. So
yeah.
in the
So
early...
Neil Phillip, Q&A box. Okay, let's see.
So Neil saying, in the early stages of the pandemic, variant spread via travel mostly was used to explain the spread of the different variants through communities as well as globally. Do you think that travel is the primary reason for variant spread, or could the same prevalence trends have been observed through processes as convergent evolution?
Okay, so if you're looking at spread of a particular lineage, right? When you're talking about a lineage, you're talking about a group of viruses that share a common ancestor. So they've evolved from a common ancestor. And if you're looking at spread of that particular lineage or that particular variant, then yes, what you're seeing is a reflection of people moving, okay? So people traveling from one place to the next and infection spreading, okay? So yes, if you look at the individual mutations that characterize those variants, so for example, you might have N5, in spike protein, N501Y. That is a mutation that you might find in several lineages and in several variants. And that has arisen, or those lineages have different ancestors, most recent common ancestors. And so that is an example of convergent evolution, where the same mutation arises in different lineages, okay? So that it's two slightly different things. Variant spreading, that's people moving. Mutations arising, the same mutation arising in different places can be due to, is evidence of convergent evolution. So I'll move down. Should I continue down?
Okay, so in the interest of time, we would save the question regarding the countries asking about information about how they can start up gene sequencing and those at the end, that both you and Gary could both give your input into that. So thank you, colleagues, for your questions. And do remember, put them in the Q&A box so our panelists could have the opportunity to look at what those questions are so we can answer them afterwards. So at this time, we shift gears and we offer a very warm Caribbean welcome, although I think it's a little bit warmer now, where Gary is, to Dr. Gary Van Domselaar. And Dr. Gary is a chief bioinformatics scientist at the National Microbiology Laboratory in Winnipeg, and also an associate professor in the Department of Medical Microbiology at the University of Manitoba. His laboratory develops methods and pipelines to understand, track, and control circulating infectious diseases in Canada and globally. His research and development activities span metagenomics, infectious disease genomic epidemiology, genome annotation, population structure analysis, and microbial genome-wide association studies. His lab has contributed to large-scale national and international genomics and bioinformatics efforts, including the Bioinformatics Workgroup of the Canadian Genomics Research and Development Initiative, interdepartmental project on AMR, the Integrated Rapid Infectious Disease Analysis Project to develop an integrated computational platform for infectious disease outbreak investigations, the Canadian COVID-19 Genomics Network, that's CanCoGen, and the Canadian Public Health Laboratory Network COVID-19 Genomics Program. And as we can imagine, Dr. Gary serves on a number of national and international scientific advisory groups, including the U.S. Centers for Disease Control and Global Coalition for Science and Regulatory Research, and Phase 4, GE. This is a very extensive body of work, Dr. Gary, and we're very honored to invite you to present to us. Welcome from our colleagues around the region and internationally. Over to you.
Okay, thank you. Can you hear me okay?
Yes, we can.
Okay, wonderful. So yes, thank you. I'm thrilled to have been invited to speak to you today on Canada's National SARS-CoV-2 Genomic Surveillance Program. Also particularly wanted to thank Dr. Carrington for her excellent presentation, including particularly the introduction, which so you've did a great job of setting me up for my presentation. I kind of didn't have a lot of time to be able to provide those introductory concepts. So your presentation actually dovetails nicely into mine. Okay, so Canada's experience in our effort for large-scale sequencing of SARS-CoV-2 began in April of 2020 through the formation of the Canadian COVID Genomics Network, CanCoGen for short, with $40 million of funding over 2 years. 20 million of that funding is directed towards the large-scale sequencing of the virus, and another 20 million has been allocated for sequencing up to 10,000 affected people in Canada. And I provided some links here for anybody who's interested in finding out a little bit more about this, the efforts of this network. Oh, and so I'm a founding member of the Virus Seek effort, and I also serve on the implementation committee. Okay. Canada is a large country, and it's also has a multi-jurisdictional public health and health network. So we work with the provinces and territories. I'm a federal employee, so we work with our provincial and territorial public health authorities collectively to address public health for the nation. When we began our sequencing effort, we had about 6 laboratories that had enough capacity to be able to do the sequencing. including my own in Winnipeg. That's this one here. Oh, I should probably change my pointer. Yeah, so this one here. Oops, nope, this one here. But since then, we've spun up another 10 sequencing centers that include some of the hospital diagnostic labs and some of our academic university sequencing partners. The central player in the network is the Canadian Public Health Laboratory Network, which I have mentioned briefly. This is where we work collectively across the country to perform the laboratory function for Canada's public health system. The National Microbiology laboratory where I work here at the top. This is sort of the headquarters for the network, but we have participation from 10 labs. Our territories, which have low populations, they partner with our provincial public health labs to carry out the laboratory public health function. Okay, so because it's a distributed network, we need to do a lot of work to centralize it. There's a lot of effort has been invested in the logistics to get the data from to get the sequences generated in across the nation, but then to get them centralized here at the national database. So the sequences are generated locally within the individual partner labs. They are standardized using what we call a data harmonizer. This is developed by our data curation group, so that the data is consistently and usefully described. Actually, our curation group has been very active in establishing international standards for the not for the sequence data, but for all of the contextual data, all that metadata, like collection date and purpose for sequencing and all the rest of that information that gets attached to the sequence data. So that's the data harmonizer takes care of. That's a technology that we've developed that takes care of that. It's uploaded then to the Canadian National Public Health Information System, which is a online web application. Is protected, so it has something what we call protected B status, which is a secret level protection. So it's designed to store sensitive information, including patient information. So it all goes into our SynFi system. It gets uploaded to the SynFi system, and then we migrate it into our national database here in Winnipeg, where I work. And we do our analysis here for the National Genomic Surveillance, all happens here. And then we publish our data to the international sequence archives like GISAID, NCBI. We also have our own portal called the VirusSeq Data Portal. Well, we just brought that up online recently, and we're still working out the process of getting the data into that portal. Essentially, we have these 3 public repositories for the sequence data that we generate. All right. Because we have so many different labs participating in the network, it's important to establish and to assess the quality of the data, of the sequence data, and that metadata that's being generated. So we have established national QA and QC specifications, and we also have an external quality assurance program where we develop proficiency panels of isolates that we send out to the partner labs, and the partner labs work up those isolates and extract the sequence data, or the nucleotide data, nucleotide material, sequence it, and analyze it and upload it to our national database, where we go and perform the analysis, and then we can assess the quality of their work. So we have 3 different levels. So there's proficient, where they're doing extremely well. Attention, if we find some areas that are are close but not quite meeting our QA/QC standards, and then corrective action required if we find any failures in any of the metrics. We've conducted 2 of these of these panel sessions so far, it takes about 6 months actually just to be able to conduct this type of quality assessment program. It's pretty involved, but it works very well for us, and it's giving us a good idea of how well we're performing for quality. We also performed a very intensive readiness assessment and recommendations for capacity building. So we knew at the start that the scale of the pandemic was going to require a lot of building of the capacity across the nation. And because we had so many of these partners, they have in different jurisdictions, they have so different levels of readiness and different trajectories for the application of whole genome sequencing for SARS-CoV-2 and for other pathogens. So we engaged in an extensive survey of all of our sequencing partners to better understand what is their operational landscape and their mandate, sort of the high level priorities of each of the labs, their current implementation status. Do they have sequencers running already or not? Some do, some don't. Some of the characterizing what are the different methods and protocols that they have in place and the infrastructure that they have in place, and some of the other more the technical things like the bioinformatics ability, the state of their workforce, and critically the data flow ownership and governance. Working in a multi-jurisdictional public health environment, the ability to share the data varies pretty wildly across the different jurisdictions. And so that gave us an understanding, like a broad understanding of the state of readiness for SARS-CoV-2 sequencing, and also allowed us to identify the critical gaps which we can invest in so that we can improve our ability and deploy that, the appropriate capacity out to all of our public health partners. And some of the biggest gaps that we did identify were specimen access and data flow, the computing infrastructure. There's not much of the really kind of advanced computing infrastructure that's required to be able to carry this out locally. We have a supercomputer at the National Microbiology Lab where we can do it on behalf of the jurisdictions, but we want to let them do it themselves. So we wanted to, so we identified that as a critical need. And then the human resources, especially in the area of bioinformatics. So, I mean, bioinformaticians in Canada are already a scarce resource. Public health bioinformaticians are like unicorns. They basically don't exist. Coming out of school, you kind of have to hire them and build that capacity internally. And so we've been very active in doing that. So this is this graph kind of shows our performance in sequencing. We've been doing the sequencing since April, but we started charting our performance since September. And so this is showing our performance here. So the graph at the top is showing the numbers of sequences that we generate per month. So and capacity building, it's not like a light switch, it's more like a dial, right? It takes time to bring the capacity online. So we deployed the capacity in the summer, but we started to see the return on our investment beginning approximately in January, where our sequence numbers increased. So from an average of about 5,000, we've in the previous calendar year, we've gotten up to about 35,000, and we're still deploying more capacity. So this is per month. So we expect that we could generate about 50,000 sequences per month, although that depends a little bit on the caseload. So at some point, you may deploy more capacity than you actually have cases. And that actually turns out to be the case. If you take a look at the bottom graph here, this is showing the percentage of sequences as relative to the case positivity rate for every month. So even though we had small numbers in September, we had a big percentage of the total cases, about 15%. As our cases went up, our percentage went way down, alarmingly low, because we were also wanted to be able to adhere to the to international guidance, like PAHO guidance, for the 5% rate. Although that's evolved, and I'll talk about that a little bit later, but the capacity that we did deploy managed to build our sequencing rate up quite significantly. So like I said, it depends on the case positivity rate, but we were managed since February to stay above 10%. Got to 92% in June as cases fell, and in July you'll see we're here at 160%. So we sequenced 160% of the cases relative to the case positivity rate for July. And so my question, well, how do you do that? And of course, the answer is that there's a lag time between the time a test is collected and the time that it's sequenced. So there's a bit of a delay. The numbers of sequences that we're sequencing in July, a big proportion of them were collected in June. Also, because we are above total capacity, we have gone and done some more of the retrospective sequencing. So going back to sequence some of the specimens that we didn't get a chance to sequence in prior months. Okay. So a lot of the capacity that we were building in the last summer and into the fall was on building up our throughput and our, but the turnaround times were, still kind of long. Time between test collection and the time of sequences generated. But we did have the agility to be able to pivot pretty quickly. So that's reflected here, and I don't have time to go through this in detail, but our ability to be able to respond quickly to the emergence of variants of concern in December of 2020 was pretty impressive. So this is a timeline in days of our response to the emergence of variants of concern. So the Public Health England investigation into the Alpha variant was launched in December 8. So that was internal. First public report to the World Health Organization was on December 14. We were able to search our database the next day to be able to confirm that we didn't have any instances of the Alpha variant of concern. The beta, which is the one that was first detected in South Africa, the B.1.351, was also reported about the same time, December 18. So we were able to search and confirm that we didn't have any instances of that. This information was used to inform our decision to suspend flights from the UK. We don't actually get flights from South Africa directly, but we do from the UK. And so we suspended our flights from the UK so they give us a little bit of time to take stock and see, to do a little bit of follow-up work to identify if we have any and how much of these variants of concern we might have in Canada. So we convened a rapid response group and we identified the priorities for identifying and sequencing these variants of concern. And within 2 days, actually Christmas Day, we identified our first Alpha variant of concern in Ontario, and then soon after in other provinces we were starting to find them using a kind of like a targeted approach where we're looking for cases of individuals with recent travel history to the UK actually any international travel history actually would be was included. And then of course we've been sequencing ever since. So, okay. So we were in a good position to be able to respond quickly to the introduction and the emergence of variants of concern in Canada through the building up of the capacity in our large-scale sequencing effort. Of course, we want to generate it's more than just generating sequences. We want to be able to use that information to perform surveillance. So we have a surveillance program, of course, in Canada for SARS-CoV-2, but it's primarily based on test results, positives or negatives. We want to bring the whole genome sequencing component, the genomic surveillance component, and combine that into our surveillance program. which we have done. So we, and this took a lot of work because you're essentially building a new surveillance program from scratch using a new technology. But it's active now and it's fully operational. So we are analyzing genomic data now, case data. We have border surveillance sequence data, and we also get wastewater surveillance sequence data from wastewater. And all of these are combined as part of our signals for our surveillance program. And then we're looking for indications of therapeutic failure, diagnostic failure, increased severity, increased transmission, vaccine escape, and immune escape. And these are all of the indicators of a variant of interest or a variant of concern. So if we do find something through our signal monitoring and it's concerning enough to affect a formal response, then we will launch an investigation. And so the Public Health Agency of Canada, where I work, this is the federal arm of that investigation. And then we would invite our provinces and territories to participate in the investigation. So this becomes a national investigation. And then the results of that investigation informs our response. So our communications and our public health policies and mitigation measures. Okay. Trying to get 10 different provinces and 3 territories to all swim in the right direction when you need a multi-jurisdictional public health environment is one of the largest challenges. And so we've had to be very active in developing national guidance. And it's not the case that all of the different jurisdictions can adhere to the national guidance, but at least it provides a reference point that for our different partner labs to attempt to adhere to. So we have sampling strategy guidance. We also have national priorities. We have a sequencing and screening strategy. So we combine genome sequencing and these like reflex screening, molecular diagnostic screens that are specific for variants of interest and variants of concern into a combined surveillance program, laboratory surveillance program. We also have conventions for naming, interpretation, and reporting. Okay, one of the I wanted to just point out a little bit more in detail about our sequencing and screening guidance. And so we spent quite a bit of time working on this, so coming in from the approach of like, what makes a good genomic surveillance program? And we were fortunate to have some valuable resources to help us in drafting our own guidance. Specifically, the European CDC has developed sequencing guidance that is very valuable, and we adapted a lot of the guidance that they provided into our own national guidance. And so that includes a component of representative or random sequencing. This gives us situational awareness. It allows us to be able to get accurate estimates of the proportions of the different variants that are circulating in our nation. There's the targeted sequencing component where we're looking for, for special populations and special settings, such as our vaccine breakthrough infections, reinfections, outbreaks in clusters, international travel, and other unusual events like super spreader events, so we can target those. We have guidance on how the sequence data needs to be tagged, and specifically the purpose of sequencing tag is a really important one, because we need to know what was the reason why you performed that sequencing. So why did you generate that sequence? Is it part of our border surveillance program, or is it part of wastewater surveillance? Is it part of a specific outbreak, or is it part of our representative sampling for estimating the proportions? So that's important to a really important field in order to be able to do useful epidemiology and surveillance for SARS-CoV-2 with genomic sequence data. There's also the timeliness of sequence generation, and that's really essential. So there is a lag between the time a test is, or a specimen is collected and it makes its way into a public health lab, and then, or one of our other sequencing partner labs, and the sequence is generated and then shared to the national lab where I work and put into our national database, and then also propagated out into the public sequence archives. And the time can range, actually. Even though it may only take, with the Oxford Nanopore technology, you can generate a sequence within a day or two. With the Illumina technologies, it may take more, could maybe take 3 to 5 days, especially because these are higher throughput sequencers. So sometimes you have to batch. So you have to collect a bunch of tests before you can send them onto the sequencer, but also just the time from getting a test, which is distributed throughout all of our clinics and hospitals and other testing centers, just the whole logistics of getting them into the labs to do the sequencing and then getting the data up into the national database adds additional lag time. So it's not the genome sequencing in and of itself is not a kind of like an active surveillance program. It's a little bit more like passive surveillance. It's about, we have approximately 2 to 3 week lag from the time a test is collected, so the time between the collection date and the time that's received to the national database, although we are continually working to try to reduce that time. We have the ability to generate enough sequence data to be effective for our genomic surveillance program, and we we're also, but the time right now that between test collection and actual sequence generation is the part where we need to invest more effort, and we are. Okay. Oh, so we meet weekly as a federal surveillance group to monitor the variants of interest, variants of concern, and other variants that we have identified that may not rise to the level of a VOI or a VOC, but are still worthwhile monitoring. So we call those track variants. And so we meet weekly and we evaluate the evidence for the different variants that are circulating in Canada using this signals assessment template here. And so the important indicators are the transmissibility, the infection severity, immunity after infection, vaccine impact, monoclonal antibody impact, and diagnostics impact. So this kind of provides a really nice synopsis of that we can use to monitor the evidence, and it may inform our decision to designate a variant as a variant of interest or a variant of concern. So Dr. Carrington showed that the World Health Organization has designations for different variants as variants of interest or variants of concern. We also have these designations in Canada. So we adopt the assessments by the World Health Organization, but it's not just we don't just blindly follow their advice. We use our own genomic surveillance program to be able to evaluate and assess the risk of a given variant that's circulating in Canada, so that we can either we will confirm that the variant has has the same risk assessment that's been designated by the World Health Organization, or we can do our own. So essentially, we will take what we will accept the assessments that they provide for different variants, but we may also, in some cases, we may have a difference of opinion, and so it's not in all cases. Essentially, what it means is that we have our own group that will provide its own expert assessment of a variant and make a decision about whether it's a variant of interest or a variant of concern. So we have our own definitions, and they're adapted from the World Health Organization definition. So this provides some of the criteria that we use for evaluating a variant. I'm not going to go into the details of it, but essentially, a variant of interest has a low bar. So it's something that we want to be able to assess and designate without too much difficulty, and it doesn't have a big impact on our public health response. If it is a variant of concern, then it does have a big impact, there is a lot more riding on the designation. So it's not just you have to be really sure that you have a variant of concern before you designate it as one. Okay. Also wanted to talk a little bit about our border surveillance program. So we implemented border surveillance and border control measures on February 18. So all of the non-exempt travelers to Canada from air and land require testing and quarantine. So we take a day 1 test and a day 8 test, and we do sequencing of all of the positive tests. This is an older table, but I wanted to use this table to point out that I think in early March when we were using our border surveillance program, we had it up and running, and we noticed a very rapid increase, like extremely rapid increase on the B.1.617, which is the Delta variant. So we were able to quickly determine from our border surveillance program that there was a problem, right? We're getting a huge influx of a new variant. It hadn't been identified as a variant of interest or a variant of concern at the time. It was just a variant called B.1.617. So that, but because of the mutational profile and what we were seeing from the borders, and also what was going on in other countries, like in India, where they were having a massive spike, that was information that we used to suspend flights from India. So while we can take stock and see, what is the situation in Canada and how are we going to be able to respond to this new variant that could be a variant of interest or a variant of concern, and indeed it was turned out to be a variant of concern, the Delta variant. Okay, again, I think I'm getting close to the end here, but I did want to point out that because we do have this large-scale genomic surveillance program in place, it allows us to be able, and we do have enough guidance on random sampling versus targeted sequencing. It allows us to be able to estimate the proportions of the variants that are circulating in Canada, which we publish. And so I have a link down here. If you're interested, you can go there and you can see for yourself what the different variants of interest and variants of concern that are circulating in Canada look like and what their proportions are. This one here in the middle, the blue one in the middle, that's Delta. This area here is accumulating data. And so like mentioned before, there is a lag time between the time that a test is collected and the time it makes it through the sequencer. So we may have numbers for this week, for a given week of maybe 4,000 or 5,000. But up here, those numbers decrease pretty rapidly. This one here, I think, in the most recent that we publicly report for the week of July 11, only 100 sequences in here. And so these can vary wildly in this grayed out area and can't be taken or used to inform public health. It's really kind of the area that's outside of the shaded area. June 20, now it's already August 11, 12, something like that. So you can see that there is quite a bit of a lag time involved in getting good numbers, stable numbers from a genome sequencing effort. And this is in Canada, where we actually have a pretty advanced effort. Oh, okay. Well, Dr. Carrington did a really good job here of describing the process of how a virus mutates as it transmits in a population, and how we can use that information to reconstruct a phylogenetic tree. So I don't need to go over this in detail. I think that was covered very well by Dr. Carrington, but I did want to just kind of give you a little bit of insight on on how we can use the genomics. So it's not just identifying the different circulating variants. It's the genome sequence data provides a lot of additional power. So we can track transmission with genomic sequence data. And the little primer that I'm showing here is illustrates how this is done. So the image at the top here, this is the reference sequence. So the first sequenced SARS-CoV-2. So it has essentially no mutations in it because all mutations are assigned relative to this original sequence over here, but that was the one that originated in Wuhan, China. So this is the virus that seeded the outbreak. Over time, the virus mutates and people will transmit that virus into new locations. So you can record a new variant in a new area of the country or in a region or wherever, but essentially the process is over time you can identify these mutations through sequencing and you can make a connection between them and start to build up a tree. So this is more of a sapling than a tree, but you know, wash, rinse, repeat as the virus continues to mutate and continues to transmit, and we perform genome sequencing on it. We can use that information to be able to reconstruct the evolutionary history of the virus and also its transmission dynamics. Of course, we get, in the end, we get these big bushy trees, which are, you know, they look difficult to interpret, but that's what our scientists are for, is to be able to use this information to be able to it is a very rich set of information that we can use to be able to track transmission within the country and also, you know, internationally. So this is a good example here of recently generated phylogeny that we used for the B.1.621. So I have designated B.1.621 as a variant of interest in Canada. We did that last week. The World Health Organization has yet to assign this as a variant of interest, and that's what I meant, that Canada has its own assessment program so that we don't just have to rely on the World Health Organization. I suspect that they will dedicate this or designate this as a VOI before too long, though. But the important point here is that the phylogeny that we generate allows us to get a sense of, like, how was it, where did it come from? So that when we know that it is prevalent in South America, specifically in Colombia, and it looks to us like that we would probably have gotten our import from South America, although there are some areas of the tree that are not showing that look like they may have introductions from Europe, which suggests that we may have multiple introductions, and we also have some evidence of a local cluster. So this is suggestive of a community outbreak and may indicate community transmission of this variant in Canada. There. Okay, there's my last slide. So in addition to being able to track the transmission using the genome sequence data and the phylogenetic trees that we build from it, we can identify these mutations that associate with the different variants of interest and variants of concern. It gives us a kind of like it's an additional signal that we can use when new variants emerge that may have the same mutations, that they may also have the same characteristics. So I'm showing this is the molecular model for the spike protein and some of the mutations that differentiate it from the reference sequence and from each other. And Dr. Carrington also alluded to this in her answer after her to one of the questions after her presentation, is that there are instances where these variants, which all have arisen in different parts of the tree, they acquire common mutations. So that is a sign of convergent evolution and something that we should be looking at, like the N501Y, which is common to alpha and beta and gamma. And these have all emerged independently, so we know that that one is important. And we can look at the different properties of the different variants to try to get a sense of what might be the biological significance of a given variant. The E484K is interesting because it's not present in alpha, but it's present in beta and it's present in gamma. And one of the distinguishing characteristics of beta and gamma relative to alpha is decreased effectiveness of monoclonal antibodies and decreased effectiveness of currently licensed vaccines. So there is good, that provides us evidence that the E484K mutation may be one of the mutations that are responsible for vaccine escape and for immune escape. And that is my presentation. I don't have an acknowledgement slide. There are hundreds of people who have been working on in our CanCogen network. So it's much too large a group for me to be able to provide any appropriate acknowledgements for. So instead, I just I decided not to provide an acknowledgement slide, but just do want to point out that this is a very large scale effort that involves participation of many of Canada's top scientists in the fields of public health and biomedical sciences and biotechnology. Okay, so thank you.
Thank you very much, Gary. Excellent presentation along with Christine, and very impressive in terms of the way you're able to explain the use of the network, the validity of having networks available, and of course we're different points in the game, but I think you were actually able through your presentation to answer some of the questions that persons had in the Q&A box as well, in terms of what are some of the considerations for getting started. And I know Christine has also been answering some of those questions. As you mentioned, what Christine and her team are doing here in the Caribbean, there are unicorns, right? And we're seeing the value of this, in terms of assessing, we need to assess the quality of the data from the labs in the network, but also in terms of, that process approach of getting the guidance. So, Gary, we want to invite you to really do stay in touch with us as you recognize from the comments and the chat. There is that interest. We have noted through our own survey from CARPHA member states that there's several countries that have already started the process. And so, we want to ensure that we have the experts that can provide the guidance. I would want us to go to the Q&A box. And so I think Giselle's question about suggestions to the countries on ensuring the quality of samples for whole genome sequencing to reduce the probability of rejection. And Gary, yes, we have our criteria in the Caribbean, but from your experience in your own network, what were some of the what are some of the critical things that you guys looked for
selecting samples for sequencing?
Yes.
Okay. Yeah. Well, right.
The quality.
Right. Okay. Well, so we do have a national specification for QA and QC, and also part of our sampling strategy. What we recommend is that we sequence samples that have a CT value of 30 or lower. So above 30, the quality of the data goes down pretty rapidly. And so we find that the rate of sequence failure, increases rapidly above a CT of 30. Below a CT of 30, we approximately 85% of the sequences that we generate end up being good sequences at CT values less than 30. So that's our cutoff for selecting samples. And then for the QA and QC specification relates partly to, what is acceptable for publication on GISAID, because they have their own QA and QC, so we take that into account. And then also, what's useful for us for, what if there's quality that's less than the quality required for deposition to the public archives, that may still be useful for us, because we might be able to get a lineage off of it, or we may be able to see some characteristic mutations or something like that. So there's an incompleteness factor is the one that I would want to point out. It's not all of the way that the sequences are generated uses a tiling approach. You kind of sequence little sections of it across the genome. Sometimes you don't, some of those little sections, the tiles will fail. And so you'll get what are called ends in that region, right? Ambiguous sequence data. So if we get above 50% sequence or 50% real data, so less than 50% ends, so 15,000 nucleotides out of 30,000 nucleotides are actual sequence data, we will accept it, but it has to be above 90% real sequence data, so less than 10% ends before we will use it for phylogenetic analysis. So we can get a lineage between 50 and 90% in some cases. We'll use it for lineage assignment, but we won't use it for phylogenetic analysis. And then I think it's a similar value for getting up onto cassette, something about 90% or higher real sequence data, less than 10% ends.
Thanks very much. And now if I can ask Christine as well, because Christine, you did give some feedback in the Q&A box in terms of persons asking for, which brand. I know that you mentioned you would have spoken about the Minayan, and then Gary also mentioned Illumina. And so, based on both here, and I invite you both to give your perspectives on that in terms of for countries starting up. Over to you.
So for us, I have to say that the existence of the Minayan was a godsend, just because it is, so accessible. It really is. I mean, as I said, it's about $3,000, $4,000 US. We had the Minayan and reagents to go with it, because so the device itself is quite cheap. The expensive part is the flow cells that you have to use every time you do a run. And but the good thing about that, we thought, was that you're sort of paying as you go. There's no big capital expenditure that we had to deal with. So I think for startup, it's very good. Now the Minayan does the Illumina still has a higher accuracy with the Minayan. But the post sequencing analysis, or the post or the bioinformatics done after the sequencing can be, and I'm sure Gary knows more about this than me, can be tailored or designed to get the best out of it. And the what is the pipelines, the bioinformatic pipelines that exist for SARS-CoV-2 are exceptionally good. And so we get very good quality sequence out of it. So to start up, I think it's great. I mentioned in the chat that one of the issues is that little tiny device has to plug into a computer, and that computer has some very particular requirements. And we had to sort of, or you couldn't just go down the road and buy it. We had to order it from overseas with the right specifications, and it took 5 months to get to Trinidad. And we ordered a second one, and it took 5 months to get to Trinidad. So that lag time is really irritating. And so if people have money, I would recommend that you get the Minayan MK1C, which has like a little onboard compute with it, which should make things easier. Although we haven't used that one ourselves yet. So there's a good starting point. 3,000, 4,000 versus 100,000 for a MySeq is it was a good start for us. We were a small project, as I mentioned.
Thanks, Dr. Carrington. Gary?
Okay, great. So I also think that so there's the Oxford Nanopore Technologies, right? Like the Minayan or the Gridiron. And then there's the Illumina Technologies. So I agree with Dr. Carrington. If you are getting started, then the Oxford is probably the way to go, because there's not a big capital investment the same way that there is for Illumina. The protocols, the Arctic protocols for doing the sequencing and the pipelines that have been developed are all excellent and really highly standardized. So there's less of a learning curve required to get up to speed. Of course, that would be the same for both for Illumina and for Oxford, but you can get up and running with the Minayan very quickly with a very low capital outlay, and you kind of pay as you go. If you want to increase your throughput a bit, then the Illumina is probably the better way to go. So, if you tend to sequence, I would say if you're going to be sequencing over 50 sequences a week, then it's time to start thinking about maybe going up to, like a grid ion will work. So that basically is just like the min ion times 4. So that will work for you as well, but the price tag goes up. So I don't know how the currencies work in your country, but in Canada, the Canadian dollar is equivalent to like an American dollar. It's about $70,000 for a grid ion versus about 3,000 or 4,000 for a min ion. Illumina has something called an iSeq, which is $35,000, and I think you can get maybe about 50 sequences a week out of it. The better option, I think, is called the mini-seq, and that's about 70,000. And then when you get up to the kind of the bigger players like the MySeq, that's about 150,000 Canadian, but the throughput is also quite a bit higher.
Okay, great. And of course, you know, we're talking about equipment as a lab and researchers, we tend to think about that in terms of ensuring that, you know, the criteria is all set as to what we're going to use this equipment for, in terms of having all those structures in place. And, you know, you spoke about, both of you mentioned the computing infrastructure that's required, and, you know, having that learning curve, even though persons might have gone through a course, let's say in bioinformatics. I guess the proof is in the pudding, and once you start actually generating your sequences and having the support of experts such as yourself, really. And here, big hint that as we see countries moving towards this, we will be reaching out in a more formal way, so that we can have you guys really see how, while you're also doing to help others that are seeking to increase their capacity as well. Because I think once this is out there, this is the way we're going to be going. Gary, there's a question here I'd like to ask you to answer about the value of the wastewater surveillance that you have in place to date.
Well, the primary value for wastewater surveillance is as an early warning signal. It's you don't get a whole genome sequence out of wastewater. You get partial genomic sequence data, but it may be enough to be able to identify a lineage. So it's a metagenomic type of approach, but its advantage is that you can see where a lineage may be surging in an area before you get it from the clinical data. So about a week in advance, we can identify that there is what appears to say be an outbreak of a certain variant in a certain community, or a town or a city by wastewater surveillance. And so then we wouldn't see that signal come in from the clinical data from the actual test collection for at least a week. And so it's a really good early warning signal. We're still in the process of just validating it and operationalizing it into our genomic surveillance program, but that's the big advantage of it.
Thanks, Gary. Okay, so Dr. Christine, over to you with this one, and then I'll ask Gary based on his own experience as well. There's a question about vaccinated individuals and the likelihood of finding a variant occurring in these vaccinated individuals. Okay, you're on mute, Christine.
Yeah, that's a question that comes up a lot, because there's this false narrative that vaccinated people generate variants. Now, anytime the virus is replicating in an individual, whether that person is vaccinated or unvaccinated, you have the opportunity for mutations to arise. If in vaccinated individuals, despite some of the headlines that you've seen, the data suggests that vaccinated people do not transmit as readily as unvaccinated people. So there's less chance of transmission, less chance of so the longer the epidemic goes on, the more you're going to get these variants. Now, the argument that is made is that the immune pressure, you vaccinate people and they produce an immune response, which then exerts an immune pressure on the virus, sort of forcing it to mutate. But the point is that whether your immune pressure is exerted by both vaccinated and unvaccinated people who get infected, whether you can get an immune response from infection or from vaccination. So that argument doesn't wash as well. And the other thing to bear in mind is that it is actually thought that these variants that we have that have, you know, lots of mutations, 15, 16 mutations in them, compared to the other ones, that those actually, it's thought that those actually arose in people who were immunocompromised. So people who have less of an immune response to an infection, to the point where the virus can remain in them for much longer and therefore accumulate mutations for over a longer period of time and then move on. So the idea that vaccinated people are going to sort of force you know, the virus to produce variants is not really, what produces variants is viral replication, unfettered viral replication. So thank you.
Thank you. Gary?
Well, yes, I agree with Dr. Carrington. As the population, as a given population becomes more vaccinated, the selective pressure is on immune escape, right? And so this is where people are thinking like, well, vaccinated people are going to create vaccine escape variants. And while that, you know, that may be the case, it doesn't take into account the whole picture, where if you have a largely vaccinated, highly vaccinated population, so the virus A, it's not going to be able to replicate as quickly or in high enough loads, so that the amount that gets transmitted is less. And its ability to be able to acquire those mutations go way down. So in the population, yes, you may, that's where the vaccine escape variants will occur, but they will occur much less frequently than they would occur in an unvaccinated or a partially vaccinated population, where you get where people who are vaccinated you may get an escape variant that arises, but if it can't transmit because the rest of the population is vaccinated as well, then it just never goes anywhere. It's where you have a partially vaccinated population that can transmit into a largely unvaccinated population. That's where the problem arises. So the solution is to get the population that is highly vaccinated, right?
Can I just add, I always like to add that all the variants we have circulating now arose before vaccination was widely available. All right, so.
Thank you very much, both of you. I think the last two questions are more or less related to the type of software that was used and a little bit more, operational in terms of can you reuse flow cells. So we can, I'm sure as you are interested in developing your own capacities, those are some of the key questions you'll need to ask as well. I know that Dr. Carrington, you shared some of, I think both you and Gary shared some of the software that's being used in your own surveillance. So once we have this recording available online through on our CARPHA webpage, which I think was shared in the chat box, and you'll receive that by email as well. We do encourage you to go back and look at the recording. Once again, a lot of valuable information that has been shared with us today. And I really want to thank both Dr. Gary and Dr. Christine for taking the time to not only share a presentation, but to answer our questions